This study is entitled Comprehensive Assessment of the Unfolded Protein Response and its Association with Tumor Progression in Pan-Cancer. The unfolded protein response UPR is critically involved in cancer development and progression. Here, we constructed a UPR gene signature to systematically assess its activity across pan-cancer, uncover UPR heterogeneity, and identify prognostic biomarkers in breast cancer. Our study has three main highlights. First, we developed a transcriptome-based UPR signature to assess UPR activation across pan-cancer. Second, we characterized UPR heterogeneity and its associations with key tumor features. Third, we identified and weighted a prognostic UPR-related biomarker in breast cancer. Based on transcriptomic data, we used WGCNA to develop a pan-cancer UPR signature and validated its robustness in independent external datasets. By comparing paired tumor and adjacent normal tissues, we found that UPR activity was significantly elevated in most tumors relative to normal tissues, indicating that UPR activation is a widespread feature of cancer. We also found that UPR activity scores were associated with multiple tumor features, including immune cell infiltration, immune checkpoint molecule expression, and stemness scores, suggesting that UPR may influence tumor progression through multiple mechanisms. Analysis of intra-tumoral UPR heterogeneity showed that UPR activity was generally higher in tumor cells, while immune and stromal cells exhibited lower UPR levels. In addition, fibroblasts displayed higher UPR activity than smooth myofibroblasts. Further analysis showed that infiltration of myeloid cells, plasma cells, and proliferating cells was significantly increased in the high UPR group, particularly myeloid cells. Further analysis of myeloid cell subsets revealed that the abundance of macro-3 and macro-4, characterized by interferon prime TAMs and lipid-associated TAMs, was increased in the high UPR. Cell-cell communication analysis further suggested that these macrophage subsets may interact with tumor cells through the MIF signaling pathway. We found that changes in tumor cell UPR scores after immunotherapy were closely associated with treatment response. UPR scores were positively correlated with immunosuppressive molecules and cytokines, and negatively correlated with immune-activating molecules, suggesting that elevated UPR activation may be linked to impaired immune function and resistance to immunotherapy. Finally, we used machine learning to identify prognostic genes in breast cancer and developed a UPR-based predictor including SHMT2, GDAR2, and SLC1. High predictor score was associated with poor prognosis and advanced stage, and the model performed well across independent cohorts. The predictor was linked to proliferation, angiogenesis, and extracellular matrix remodeling, supporting its clinical relevance. Overall, this study systematically evaluated UPR activity across pan-cancer, revealing its intertumoral and intratumoral heterogeneity and its associations with key genomic and immune features. We also identified a UPR-related prognostic biomarker in breast cancer with potential clinical value, providing important insights into the role and therapeutic potential of UPR in cancer.